I want you to try something right now. Just hold up your hand and look at it.
Like right now, as you're listening.
Yeah, exactly. Place your fingers, look at your skin, check out your fingernails. I mean, it feels completely solid, doesn't it? It feels static.
Right. It feels like nothing is really happening.
Exactly. But under the surface, like right at this very second, there was this frantic, highly choreographed, microscopic dance happening just to keep you intact.
Oh, it is an absolute frenzy of activity. We're talking billions of moving parts. Molecular machines literally tearing themselves down and rebuilding.
And we just walk around completely oblivious to it.
Totally oblivious, yeah.
And so that invisible frenzy, that is our focus today. We are doing a deep dive into a massive stack of biological and medical literature.
Comprehensive manuals, really, on cell structure, reproduction, developmental biology.
Right. But our mission here isn't to just memorize a textbook for you. We want to unpack the sheer elegance of this machinery. Like, how does a single microscopic cell in your finger know exactly when to copy itself to heal a paper cut?
Mm-hmm.
And how does an entirely different biological system manage to shuffle the genetic deck to create, brand new, totally unique humans?
Because when you zoom out and look at the whole picture, the copying, the dividing, the shuffling, these microscopic events aren't just trivia.
Right.
They are the fundamental basis for growth, for tissue repair, and, well, ultimately for the entire engine of evolution.
Okay, so before we can even touch on making new people, we have to figure out how we maintain the bodies we already occupy, right? Because we are constantly shedding skin, replacing blood cells, healing scrapes.
Yeah, and to do any of that, cells have to divide and multiply. But the sheer logistical challenge of cell division it's staggering. You have to consider the packaging problem of DNA.
Okay, the scale here completely blew my mind. If you took the DNA from just one single actively dividing cell, unspooled it, and laid it out end to end.
Just one cell.
Just one. It would stretch the length of an entire football pitch.
Yeah.
Inside one microscopic cell. And you have, what, trillions of them?
Yeah, trillions. You have this incredibly long, fragile, 2-meter double helix of genetic code crammed into a space a fraction of a millimeter wide.
It's insane.
It is. And you can't just slice a cell down the middle and hope the DNA sorts itself out.
Right, because the strands would snap.
Exactly. It would be a catastrophic, tangled mess.
I was trying to picture how that even works, and it's a lot like moving out of a house.
Yeah.
You can't move a giant, loose pile of yarn across town without it knotting up.
Oh, that's good.
Yeah, you have to pack it tightly into boxes first.
That is the perfect way to visualize it, honestly.
Yeah.
The cell's version of packing boxes relies on these positively charged proteins called histones.
Histones, right.
Because DNA has a negative charge, it naturally wraps tightly around these histone proteins, and that forms dense little clusters known as nucleosomes.
So it's basically like wrapping thread around a wooden spool.
Yeah, exactly like a spool.
Yeah.
And those spools don't just sit there, they coil up even more.
They supercoil, right?
Right, they fold back on themselves again and again until they form those iconic, dense, X-shaped structures we call chromosomes.
Which is the only time DNA is packed that tightly, right?
Yeah.
Like the only time chromosomes actually become visible under a microscope is when the cell is actively preparing to divide.
Precisely.
Which, brings up something I struggle to wrap my head around. A cell doesn't have a brain.
No.
It doesn't have a central nervous system. So how does it actually know when to divide? I mean, it's not just randomly splitting whenever it feels like it.
Not at all. It follows a very strict timeline called the cell cycle. It is heavily, heavily regulated.
Okay.
In fact, a cell spends the vast majority of its lifespan in a phase called interphase. And historically, biologists called this the resting phase.
Which is a terrible name, right?
Terribly misleading, because
Anyway.
the cell is doing everything but resting.
It's prepping for the main event.
Yeah.
Because during interphase, the cell goes through distinct stages, right? G1,
Yeah.
S, and G2.
Yeah. Think of G1 as the initial growth phase. The cell is gathering materials, making enzymes.
Just getting ready.
Right. Then it hits the critical S phase, or synthesis phase. This is the moment the DNA actually replicates.
Wow.
Every single chromosome copies itself to become double-stranded, so that you eventually have enough blueprints for 2 separate cells.
And then G2?
G2 is the last-minute prep, synthesizing the specific organelles and proteins needed to physically tear the cell in two.
Okay, but the question remains, what actually pulls the trigger to make that happen?
Chemical checkpoints. And the keys to passing these checkpoints are small, constantly fluctuating proteins called cyclins.
Cyclins.
Yeah. As a cell progresses through interphase, these cyclins build up. When they reach a certain concentration, they physically bind to specialized enzymes called cyclin-dependent kinases, or CDKs for short.
Okay, so the literature mentioned that this cyclin CDK complex phosphorylates other proteins.
Yes.
Which sounds intensely complicated, but if I'm understanding the chemistry here, it basically means it attaches a phosphate group that changes the target protein's physical shape.
You've got it. And in biology, shape is everything.
Because it dictates function.
Exactly. When you change a protein's shape, you act like a molecular light switch. You are suddenly turning that protein on or off.
Oh, wow.
So when these CDKs change the shape of target proteins, it kicks off this massive cascade of reactions that pushes the cell past the point of no return.
Okay, so we've passed the checkpoint. The moving boxes are packed. We enter mitosis, the actual active division. And there's an acronym here, PMAT, right? Prophase, metaphase, anaphase, telophase.
Right, PMAT. Let's look at the physical reality of what happens next. In prophase, the nuclear membrane, the vault holding all this packed DNA, actually dissolves.
Wait, it just melts away.
It breaks down, yes, so the chromosomes are just floating free.
And then comes metaphase, which is almost strangely orderly, reading about it. All those supercoiled chromosomes line up perfectly single file right along the equator of the cell.
It's a very striking image under a microscope.
I bet. And these tiny structures called spindle fibers reach out and hook onto the center of every single chromosome.
They are like microscopic cellular fishing lines. They anchor to the chromosomes and they create immense physical tension.
Because they're getting ready to pull.
Exactly. Because in the next phase, anaphase, those spindle fibers start to contract. They literally pull the duplicate chromosomes apart, dragging the halves toward opposite poles of the cell.
I could just imagine the sheer mechanical force happening there. It's crazy.
It really is. And finally, telophase. The DNA halves have safely arrived at opposite ends of the cell. New nuclear envelopes form around them to protect them, and the cell executes cytokinesis.
The final snip.
Right, the final snip where the outer membrane pinches inward, like drawing a purse string closed, creating two entirely separate self-contained cells.
The precision is just astonishing. And the reason this meticulous, error-free process matters so much is the end result. Mitosis produces perfect clones.
Yes, it is asexual reproduction at the cellular level.
Which makes sense, right? Like, if I scrape my knee, I don't want my body experimenting with new, unique skin cells. I want the exact same skin cells that were there yesterday, perfectly patched.
Exactly. Without perfect, identical replication, complex, multicellular life just fails.
Yeah.
You couldn't grow a single millimeter, and you couldn't survive a paper cut.
Okay, so mitosis is the ultimate tool for stability. Exact copies. But stability has a massive biological blind spot.
A huge one.
Right, because if the environment changes drastically, say, an ice age hits, or a completely novel virus sweeps through the population, a group of exact clones is incredibly vulnerable.
Sitting ducks.
Yeah, if the virus can kill one of them, it can kill every single one of them instantly.
And that vulnerability is the fundamental flaw of relying purely on asexual reproduction. Clones are great until the world changes.
Which it always does.
Always. Biology had to invent a loophole to introduce diversity, to make sure a population wasn't just a biological monolith. That loophole is the second massive mechanism we need to look at, meiosis.
Shuffling the genetic deck.
Right. Meiosis is the specialized process of creating gametes, sex cells. And the math completely shifts here.
How so?
Well, in mitosis, you create cells that are diploid, meaning they have the full, normal set of chromosomes.
Two sets, one inherited from your mother, one from your father.
Exactly. But meiosis takes a diploid cell and divides it twice. It deliberately reduces the genetic material to create haploid cells.
Haploid, meaning half.
Yes. Cells that contain only one single set of chromosomes, half the usual amount.
Okay, I have to play devil's advocate for a second here.
Go for it.
Looking at this purely from an energy perspective, asexual reproduction like cloning is brutally efficient.
Oh, absolutely.
You don't need specialized reproductive organs. You don't need to spend massive amounts of calories trying to find a mate, impress a mate, and combine genetics. You just divide and conquer. So why on earth did biology bother evolving this incredibly complicated, energy-draining, highly risky process of sexual reproduction?
Because it's the ultimate evolutionary trade-off. The biological cost of sexual reproduction is immense, you're right. But the reward is genetic variation.
Variation.
Variation is a species' ultimate survival insurance policy. If every offspring has a slightly different genetic makeup, well, some might naturally resist that new virus.
Right.
Some might naturally tolerate colder temperatures. It provides the raw material that natural selection acts upon to keep a species alive over millions of years.
But how does meiosis actually guarantee that variation? Because, like, it's not just a blind grab bag where a cell randomly halves the chromosomes.
Yeah.
There is active, deliberate genetic mixing taking place.
It happens through two beautifully elegant mechanisms.
Okay.
The first is called crossing over or recombination. During the very first phase of meiosis, the homologous chromosomes, meaning the matching pair, one from your mother and one from your father, they physically align with each other side by side.
And they don't just sit there. They literally break apart and trade pieces.
Yeah, it's pretty extreme.
Mhm.
Large multi-enzyme complexes physically cut the DNA strands of the maternal and paternal chromatids at specific overlapping points.
Called chiasmata, right?
Yes, chiasmata. They snip them, swap the ends, and glue them back together.
So you are basically stitching together traits from your parents to forge brand new hybrid chromosomes that has literally never existed before in the history of the world.
Never. It's entirely new, and that's just step one. The second mechanism is independent assortment.
Right. Okay. This is when the newly mixed chromosomes line up along the equator of the cell to be divided, sort of similar to metaphase in mitosis.
Right.
But the catch here is that they line up completely randomly. The maternal version might face left, the paternal version might face right, and it's a coin flip for every single chromosome pair.
And the math on this coin flip is just staggering. Because human cells have 23 pairs of chromosomes sorting randomly, just one single parent can produce over 8.3 million possible genetic combinations in their haploid sex cells.
See, I need to let that number sink in. 8 million combinations from one person.
Just one.
So when you multiply your 8 million potential combinations with your partner's 8 million combinations, that is over 64 trillion possible genetically distinct humans that you two could theoretically create.
Yep, 64 trillion.
64 trillion. And yet somehow the universe landed on this specific combination that made you.
It really reframes how you look at the concept of being genetically unique, doesn't it?
It really does. And speaking of genetic uniqueness, I actually found this fascinating tangent in the literature.
Oh, yeah?
Yeah, humans play by strict rules. We are diploid. We have 2 sets of chromosomes. End of story. If a human gets an extra set, it is completely fatal to the embryo. But the plant and animal kingdoms don't always follow that rule.
You're referring to polyploidy.
Yes. Having multiple complete sets of chromosomes, like potatoes, for example, are polyploid. And certain fish, like specific species of salmon, can have up to 400 chromosomes.
400.
It's wild. It's a wonderful reminder that biology is an endless laboratory.
Yeah. In plants, polyploidy often leads to larger cell size, bigger fruit, increased hardiness. It's really just another flavor of that genetic variation we were talking about.
Okay, so meiosis successfully creates these unique haploid half sets of DNA. But a haploid cell is utterly useless just sitting there on its own.
Right. It can't do anything.
It needs to be safely delivered and combined with another half set. And when you look at the female and male delivery vehicles, the gametes, they evolved completely opposite survival strategies.
They really represent two inverse engineering solutions to the exact same problem. Let's take mammalian gametes.
Yeah, if you look at the diagrams, the male gamete, the sperm, is built like a minimalist, stripped-down speedboat.
Good analogy.
It carries 0 excess baggage. It's literally just an engine and a payload.
It's often summarized in biology as the many motile strategy.
Motile meaning it moves, has a tail for propulsion. The middle section is absolutely jam-packed with mitochondria, the powerhouses, which provide all the ATP energy required to whip that tail.
Right.
The head contains the highly condensed genetic payload. And right at the very tip, there is this microscopic warhead of enzymes called the acrosome.
Okay, now contrast that racing boat with the female gamete, the ovum. It is a heavily fortified, fully stocked mothership.
The few, fat, fixed strategy, it doesn't even have a mechanism to move on its own.
It doesn't need to. Form follows function perfectly here. The ovum's job isn't to travel. Its job is to provide all the initial resources for a developing embryo.
It's huge, right?
It is massive compared to the sporm, packed with lipid droplets to serve as long-term food reserves.
Plus, it has some serious armor.
Oh, absolutely. The zona pellucida, it's a thick, highly protective, jelly-like glycoprotein layer surrounding the main cell membrane.
And this dichotomy isn't just an animal thing. Flowering plants have evolved their own highly specialized delivery systems that are arguably just as strange.
Plant reproduction is wonderfully complex. The male gamete is housed within a pollen grain. But a single pollen grain doesn't just have one payload, it contains 2 distinct haploid nuclei.
Right. A generative nucleus and a tube nucleus.
Exactly. While the female gamete sits nestled deep, securely hidden inside the plant's ovary within a structure called an ovule.
So the delivery vehicles are built, they're launched. Now comes the exact moment they meet, fertilization. And understanding the actual biochemistry of this moment, I mean, it plays out exactly like a microscopic bank heist.
It really is a high-stakes, highly secure break-in. In mammals, the sperm swims up and finally reaches the ovum, but it immediately slams into that thick armor we mentioned, the zona pellucida.
The bank vault wall.
Right. And this is where that warhead at the tip of the sperm activates. It's called the acrosome reaction. The sperm literally bursts its acrosome open, spilling digestive enzymes onto the jelly layer to chemically burn a tunnel through the armor.
But the heist doesn't end there. Once a single sperm makes it through the tunnel and its membrane touches the actual inner cell membrane of the egg, the bank instantly drops titanium blastors.
It's a cortical reaction.
Yes.
It happens in an absolute split second. Just beneath the surface of the egg are these structures called cortical granules. The moment the sperm fuses, these granules dump their contents into the surrounding space.
Changing all the locks on the vault.
Exactly. They destroy the sperm binding sites so no other sperm can attach, and they instantly harden that outer jelly layer into an impenetrable wall called the fertilization membrane.
Which begs the question, why the intense security? Like, why not let a few robbers in?
Because if a second sperm manages to fertilize the egg, a condition called polyspermy, it's disastrous.
Oh, really?
Yeah, you would have a zygote with 3 sets of chromosomes. In human biology, having that much excess genetic material completely breaks the cellular machinery. The chromosomes can't pair up properly during the next division. It is a fatal crash.
Wow.
So the cortical reaction ensures only the absolute fastest, most capable sperm delivers its payload.
Okay, I'm curious about the plants, though. You mentioned earlier that the pollen grain carries 2 nuclei. Are they pulling off a double heist?
Essentially, yes. It's a process unique to them called double fertilization.
Oh, okay.
When a pollen grain lands on the receptive part of a flower, that first nucleus, the tube nucleus, acts like a demolition crew. It controls the growth of a long pollen tube that physically digests its way all the way down the style and into the ovary.
It bathes the tunnel.
Yes. Once the tunnel is clear, the second nucleus, the generative nucleus, travels down it. Along the way, it actually divides into 2 male nuclei. The first one fuses with the female egg cell to form the actual zygote, the baby plant.
And the second one?
The second one fuses with 2 separate polar nuclei that are already waiting inside the ovule. This fusion creates a strange triploid cell, meaning 3 sets of chromosomes, which develops into the endosperm.
The endosperm. That's basically the packed lunch for the seed, right? It's the starchy food supply that keeps the plant embryo alive underground before it can sprout leaves and start photosynthesizing.
Exactly that. Looking at both plants and animals, what is so striking is the exquisite evolutionary timing required for all this.
It's not random.
Not at all. The female structures aren't just passive targets. They are actively guiding, testing, and screening the male gametes, putting up extreme physical and chemical barriers to ensure only the most fit genetic material makes it to the finish line.
We've painted this beautiful picture of elegant, flawless machinery. The cell cycle humming along perfectly, meiosis shuffling the deck with precision, the high-stakes timing of fertilization. But these systems are physical. And physical things break. What happens when the microscopic machinery stalls out or just ignores the rules entirely?
Well, that intersection is exactly where biology becomes medicine. Let's look back at mitosis. We talked about how tightly regulated the cell cycle is, controlled by those cyclins and the CDK checkpoints. They are the traffic lights of the cell.
But sometimes a cell decides to run a red light.
Unfortunately, yes. When those genetic control mechanisms mutate or fail, a cell begins to divide uncontrollably. It just keeps repeating the cell cycle over and over, piling up clones without stopping.
Wow.
That unchecked division is the fundamental biological definition of cancer.
Which brings up a really elegant mathematical tool doctors use, discussed in the literature, called the mitotic index.
Yes. It's a remarkably simple but crucial diagnostic ratio. The mitotic index is calculated by taking the number of cells in a tissue sample that are actively caught in the middle of dividing somewhere in PMA and dividing that by the total number of cells in the sample.
Okay, so in a healthy slide of tissue, you'd expect a pretty low ratio, right? Because like we established, most cells spend their time chilling in interphase, just doing their normal daily jobs.
Exactly. But if a pathologist calculates the mitotic index of a biopsy and the resulting number is unusually high, it tells them that an abnormal proportion of those cells are frantically dividing. It is a massive red flag indicating malignant, cancerous tissue.
The brilliance of it is that oncologists can use that exact same math to see if a treatment is working.
Right.
If you give a patient chemotherapy and their tumor's mitotic index plummets over the next few weeks, it proves the drugs are successfully hitting the brakes. They are forcing those runaway cells out of mitosis and trapping them back in interphase.
It is a perfect example of how understanding a microscopic mechanism, the cell cycle, allows us to intervene and save lives on a macro level.
Which brings us to another major medical intervention. Not with uncontrolled division, but with a failure of fertilization. We talked about the incredible bank heist the sperm has to pull off.
Right.
But what if the delivery vehicles just aren't up to the task?
Severe male infertility is a significant roadblock for many couples. To put the heist in perspective, a healthy mammalian system is launching roughly 20 million sperm per cubic centimeter of semen.
20 million.
But it's not just a numbers game. It is heavily dependent on motility. Around 50% of them need to be able to swim vigorously and in a straight line.
So if they're swimming in circles, or if they're structurally abnormal, or if there just aren't enough of them in the payload, they can never reach the egg, let alone chemically burn through that thick zone of pellucid armor.
Exactly. The natural fertilization process just stops cold.
And for a long time, biological limits meant couples facing this had very few options. But reproductive medicine developed a massive breakthrough in IVF technology known as ICSI.
Intracytoplasmic sperm injection.
Intracytoplasmic sperm injection. It's exactly as direct as it sounds. Doctors harvest a single, healthy-looking sperm. They don't make it swim an obstacle course. They don't require it to perform the acrosome reaction to blue torch its way through the jelly layer.
Nope.
They use an incredibly fine microscopic glass needle to physically puncture the egg and inject that single sperm directly into the cytoplasm.
They completely bypass the heist.
They just teleport the bank robber straight through the titanium doors and into the vault.
It's wild. It removes all the natural physical barriers to conception.
Yeah.
ICSI is now used in at least 30% of all IVF treatments globally. It offers profound hope to couples who previously had zero biological chance of conceiving.
It's an incredible medical triumph. But, and the sources touch on this, it makes me wonder, if we are entirely bypassing the egg's intense natural screening process, like we aren't making the sperm prove it's the fastest, or that it has the chemical enzymes to breach the armor, does that change the evolutionary fitness of the resulting embryo?
You're hitting on a deeply complex question, one that bioethicists and geneticists discuss extensively. You are absolutely right that ICSI completely circumvents the harsh, microscopic natural selection that normally occurs during the race to the egg. We are purposefully prioritizing the immediate human desire to create life over the ancient, ruthless screening mechanisms of evolution.
Wow.
It is one of the most profound examples of human science actively overriding biological limits.
It really puts into perspective how much power we've gained just by mapping out how our cells operate. Let's take a breath and recap the sheer scale of the journey we've been on today.
We have definitely scaled from the molecular to the human.
We really did. We started by looking at a single cell, watching it spool 2 meters of DNA onto tiny protein spools, perfectly cloning itself through the mechanical tension of mitosis just to heal a scratch on your hand. Then we watched meiosis take that exact same biological machinery and twist it, deliberately breaking and swapping chromosomes to shuffle the genetic deck and guarantee that life can adapt.
And we saw how those highly customized half sets of DNA are weaponized into specialized gametes, culminating in the high-stakes, heavily armored biochemical break-in of fertilization.
The exact sequence of billions of microscopic events that resulted in you, uniquely you, listening to this deep dive right now.
But before we close out, there is one final, almost mind-bending biological reality buried in the margins of these texts that completely changes how we relate to our own bodies.
Okay, I love a good mind bender. What is it?
Well, we have spent this entire deep dive focused on the cell cycle. We've talked about constant division, frantic regeneration, checkpoints, and turnover. But there are exceptions. Biology has what we call permanent cells.
Cells that don't divide.
Right. Once they are fully formed during your early development, they enter a phase called G0. They exit the cell cycle entirely. They never divide again.
Wait, never. Like for your whole life?
Never. The specialized nerve cells in your brain that are currently firing to process my words.
No.
The cardiac muscle cells that are physically contracting to keep your heart beating right now. The transparent cells forming the lens of your eye. They do not replace themselves. They are as old as you are.
Wow. So while my skin is constantly replacing itself every few weeks, my heart and my brain are just, they're the original hardware.
They are the original biological hardware you were born with. They don't refresh. They are experiencing the entire, uninterrupted, decades-long journey of your life right alongside you, from day one until the very end.
That is deeply profound. Next time you look down at your hand, just think about that. Think about the billions of cells frantically, violently tearing themselves in two to maintain your shell, the ancient evolutionary lottery that created your blueprint, and those few permanent cells quietly keeping the rhythm of your life start to finish.
It's quite a lot to think about.
It really is. Thank you for joining us on this deep dive into the microscopic world. Keep wondering, keep exploring, and we'll catch you next time.